How is ATP generated? The same way it was in glycolysis, by substrate level phosphorylation. Okay, your input here, putting in two acetyl groups attached to coenzyme A. We got this, remember, from the two previous acids. You don't need to worry about the rest of this. So basically, you're just putting in two acetyls hooked onto coenzyme A, CoA. Output, and again, concentrated energy storage molecules. You do produce waste products, CO2. And again, we're talking about two passages, 2 ATPs, 6 NADHs, and 2 FADH2s. Electron transport, again, this occurs in the mitochondria, but it requires a membrane because that's where electron carriers are embedded. So it's the inner membrane of the mitochondria. How is ATP generated? Chemiosmosis, which remember is the mechanism of oxidative phosphorylation, that process I described earlier. Complicated, but very important because that's where most of the ATP is going to be generated. What's the input? The input, electrons. High-energy electrons from NADH and FADH2. And from 10 NADHs and from 2 FADH2s. Now you know we got the 2 FADH2s with 2 passages through the Krebs cycle. How can you account for 10 NADHs? How much did you make in glycolysis?
Glycolysis,
2. Okay, for each glucose. How much was made in the Krebs cycle, NADH?
6.
6, that's 8. Where are the additional 2 coming from?
From the bridge transition.
From the breakdown of pyruvic acid to acetyl. Remember you got 2 ATPs there, each one. You got 2 pyruvic acid, you end up with 2 acetyl, hooked on the coenzyme A and shoved in the Krebs cycle. You got 2 ATPs, or 2 NADHs there. That accounts for 10. So electrons from these compounds are being put into electron transport, and then our summary of ATP, our total, well, for each NADH inserts electrons, we get 3 ATPs. We inserted from 10 times 3, 30 ATPs. For each FADH2 that inserts electrons, you only get 2 ATPs, so 2 multiplied by 2, 4. So our total is 34 ATPs. How much ATP we get in glycolysis? Net? Each glucose?
2.
2. How much in 2 passages of Krebs cycle? 2 again. 34 here. So without a doubt, you can see this is where most of the ATP is made. And this requires oxygen in our cells. Okay? Aerobic cellular respiration. You can see without the Krebs cycle, and especially without the electron transport, we don't make much ATP. We simply don't have enough energy available for cells to sustain themselves. That's why we need oxygen. The cell can generate a lot more ATP. As a result, have a lot more energy available to do its work. Very important. And this stuff is going on all the time. Even when you're at rest, your cells are metabolizing actively. You're generating ATP. Okay, so the summary then, 2 ATPs net from glycolysis, 2 from 2 passages through the Krebs cycle, 34 from electron transfer gives a total of 38. Now in bacteria, that's what usually happens because there are no mitochondria. But we got a problem. We generated NADH, 2 of them, in glycolysis. And where does glycolysis occur in the cell? In the cytoplasm. But where does electron transport occur? Within the mitochondria. The problem is this. NADH is too large a molecule to pass the mitochondrial membrane. So instead it shuttles its electrons across. They're picked up by another molecule. If it's like NADH and inserts electrons right at the beginning of the chain, electron transport chain, you get your 3 ATPs. If it's like FADH2, it's going to insert them farther down the chain, you're only going to get 2 ATPs, and that's often the case. So in most cells, we subtract those 2 ATPs because the hydrogen electrons are being transferred from NADH to an FAD-like molecule, and we lose an ATP for each transfer. We transfer 2, we lose 2 molecules. So the total here now then would be, for most of our cells, 36 ATP. So if you ask what's the total ATP production on average, it's about 36 ATP for each glucose, with a few exceptions, which I listed here. Cardiac muscle cells, liver cells, and kidneys. Okay? There, they have a carrier like NADH. There's no ATP lost, and you still get the 38 on average, okay? And if you look at them, cardiac muscle, kidney, liver cells, highly metabolically active cells. They require a lot of energy, so it's not surprising. They got a different carrier, and they're still losing ATP, more efficient. But skeletal muscle, for example, neurons, and most of the cells in our body, it's 36. That's the magic number. Again, it's an average. Okay? But there are the exceptions. Like almost anything in life, there are always exceptions. Okay, so the overall reaction, you see right here, C6H12O6, that's the general molecular formula for a 6-carbon sugar like glucose, 6 oxygen, 4 ATPs, you get 6 carbon dioxide, 6 waters, and your energy, most in the form of heat, about 60%, the remaining 40% or so, 36 ATP. Okay? And again, that's pretty good efficiency. Better than any human-built energy or engine I'm aware of. Okay? You get that kind of efficiency in your engine, I'd like to know what you're driving. You just making engines efficient. And the reason that our cells break the glucose down in stepwise fashion, one reaction after another, small reactions, is to release energy in small amounts, to trap more in energy storage molecules like ATP. Otherwise, if you took the glucose and just blew it apart, carbon dioxide and water, most energy would be in the form of heat, and it'd be wasted. We get very little energy that we could use, or cells could use. Okay, and lastly then, a little bit about lactic acid fermentation. This is when our cells carry on for brief periods of time when they don't have the oxygen available to them to carry on aerobic cellular respiration. And certain cells can do this for a limited period of time. When you were studying skeletal muscle metabolism, I'm sure you talked about lactic acid fermentation. Okay? And here you can see it's anaerobic, which means it does not require oxygen. Organic molecules oxidize. Again, you're transferring these high energy electrons. And some of the energy again from those electrons is going to generate ATP. And in this case, your end product and final electron acceptor is not oxygen, it's lactic acid. And as I said, there are a number of fermentation reactions. They're named for the organic molecule that's at the end, the final electron acceptor. For example, alcohol fermentation, which some microorganisms carry on. Very important to us if we like alcoholic beverages, because that's where we get our ethyl alcohol. Okay? For alcohol fermentation. In that case, what they do is they break down the sugar, break it down into a molecule, and then that molecule then is converted to alcohol instead of lactic acid. Okay, the first stage in most fermentation reactions is simply glycolysis again. You're breaking down the sugar, glucose. Again, you've got your 2 groupic acids forming, just like we saw before. A net of 2 ATPs, and again, the 2 NADHs, like we saw before. Then the 2 groupic acid molecules are reduced using energy from NADH to form 2 lactic acid molecules, the end product. Now, most of the energy from the glucose, of course, is in the end product, which is lactic acid. So what are you left with as far as energy storage molecules? 2 ATPs, and that's it. Because we use energy from the NADHs to convert the groupic acid to lactic acid. So for each glucose, you're only getting 2 ATPs. How much did you get aerobic cellular respiration? ATPs? 36 in most cells, and in some cells, 38. So you can see, much less efficient process. The beauty is you don't need oxygen, but the downside is you're not going to make much ATP. You're not going to have much energy available. And that's why our cells, some of them anyway, can carry on lactic acid fermentation, but only for brief periods of time. It's like when you're running a race, okay? A lot of power for those skeletal muscles. You can do a sprint and barely breathe. At the end, of course, you hit the wall and you're panting trying to recharge the oxygen. If you're going to run a longer race, like a marathon, you've got to be breathing regularly to generate enough ATP to keep that muscle activity going. So again, in this process, no Krebs cycle involved, no electron transport occur, and that's why you're stuck with only the 2 ATP that were generated in glycolysis. No oxygen used, but it can occur when oxygen is present, it just doesn't use it. Okay? In our bodies and those of most other animals, this is what we see. When they don't have oxygen available, they occur in lactic acid fermentation. As I said, some microorganisms that can metabolize under anaerobic conditions can carry on this as well. And again, the summary of lactic acid fermentation, which is already clear now. You can see it occurs in the cytoplasm because we're talking about glycolysis. Again, ATP generation, substrate level phosphorylation. Input, 1 glucose, 2 ATPs to keep glycolysis going, and then energy from 2 NADHs. So we use those molecules. The output then, 2 lactic acid molecules, a net of 2 ATPs, and again, you've lost the NADH because it provided the energy to convert the pyruvic acid to lactic acid. So all you have here is an NAD. So your energy storage molecules, 2 ATPs net. And that's it for each glucose. Okay. Any questions? Yes.
Can't the heart and the liver use lactic acid?
I'm sorry?
Can't the heart and the liver use lactic acid?
Yes, now cardiac muscle can use lactic acid as a fuel source, but most cells can't. Okay, like skeletal muscle can't, and a lot of other cells, neurons and so on. Cardiac muscle is unique in that regard, but the problem with cardiac muscle, it can't carry on lactic acid fermentation, okay, because it can use the lactic acid, but it needs more energy. And that's why it carries on aerobic cell respiration, why you have to have a good supply of cardiac muscle to supply the oxygen. Not so much the fuel is the limited source, it's the oxygen. Okay? Okay, that concludes our coverage then of this chapter. We've already covered the respiratory and digestive system. So you have now material for next week's exam, which is a week of